Okay, hi everyone. This is a lecture for part one of chapter three. I'm going to break it up into two parts just because it's kind of long. So I'll probably do this one for about a half an hour, and then the next one might be slightly longer, about 45 minutes or so. Okay, so our last chapter, chapter two, was just an intro to basic chemistry, and we sort of talked about the formation of different types of bonds, right? And then when we're going to form bonds between atoms or between elements, we then can make molecules, okay? And so this chapter focuses on the chemistry of organic molecules, okay? So super important chapter. Again, we're always focusing on organic molecules because these are the molecules that you're going to find in living organisms. Um, just like the last chapter, I kind of started the chapter with the little introductory paragraph that you find in the textbook. Okay, and so the little introduction kind of, again, it's always the sort of trying to capture your interest and sort of why should you care about organic molecules, right? So this is talking about trans fats and cardiovascular health. And so trans fats we've probably heard of before, and you can actually see them on the package, and there's lots of warnings about eating trans fats now. Okay, so it's a type of fat or a type of lipid, and we're going to talk about fats and we're going to talk about lipids later on in the chapter. And so these trans fats are made by adding hydrogen to unsaturated fats, right? So there's a process called hydrogenation, and so basically, if we add hydrogens to unsaturated fats, we hydrogenate them, and basically it's a way of increasing the shelf life of foods. However, those can sort of then become these trans fats that are believed to be actually worse than regular saturated fats in the diet. Okay, so these trans fats are, think about them as being sort of man-made or chemically altered, right, in order to increase the shelf life of a product, like I said. All right, so anyway, it is required that foods that contain trans fats are labeled because of the adverse health effects that they may cause. All right. So as I said, we will talk about lipids and we'll talk about fats, which are a type of lipids later on in the lecture. As you can see here, this is the breakdown. We'll talk about organic molecules in general. Then we'll talk about the four different types of biomolecules, four different types of organic molecules, right? Main groups: carbohydrates, lipids, proteins, and nucleic acids. All right. So again, to just start off in general, talking about organic molecules, what are organic molecules? They are molecules that contain carbon and hydrogen, and they may contain other atoms as well or other elements as well, but organic molecules contain carbon and hydrogen. There are four main classes of biomolecules or organic molecules. I'll usually refer to them as biomolecules that exist in living organisms. Okay, and I just mentioned them in the outline there: carbohydrates, lipids, proteins, and nucleic acids. Obviously, yes, you should know the four main classes of biomolecules. They are listed here. Okay. All right, so this is just giving you kind of just some examples of um, some different types of biomolecules, particularly, actually, you know what? One second. I got to look at this figure. I always forget exactly what they're referring to here, but it's basically meant to be sort of an illustration of some different types of biomolecules. One second. Yeah, so as I said, this is kind of meant to illustrate these four different types of biomolecules, right? So if you look at panel A, so that's a canola plant. And so it's drawing your attention to lipids, right? Canola oil, lipids that obviously store energy in the plant. Here is illustrating sort of carbohydrates, right? Structural carbohydrates that are present in the tree. So basically, structural carbohydrates provide structure to plant cells, to the cell wall. Okay, in panel C there, right? Proteins that form hemoglobin. So hemoglobin is a protein. Hemoglobin is a pretty complex protein, and it carries oxygen around on your red blood cells, right? So that's what that is illustrating there. And if we look at panel D, it's basically just kind of showing you the lioness and her offspring here, and that obviously nucleic acids, right, are your molecules that essentially are going to make up DNA and RNA, and that gives her the ability to pass down traits to her offspring. Okay, so each one of those pictures is just a little bit illustrating one of those different types of biomolecules that I mentioned on the previous slide. All right, now, again, I told you we're focusing on organic molecules. If we compare and contrast, or sort of mostly contrast organic molecules versus inorganic molecules, again, remember, organic molecules contain carbon and hydrogen. These are molecules that we find in living organisms, hence why we will focus on these molecules in biology, okay? So again, you can see inorganic molecules, they usually contain positive and negative ions, right? Usually use ionic bonding, so like sodium chloride, right? Um, for organic molecules, always contain carbon and hydrogen, always use covalent bonding. They're often quite large with many atoms. These guys in inorganic molecules are often smaller, and these are often associated with non-living matter, and these with living, okay? All right. So since we said organic molecules are composed of carbon and hydrogen, obviously we have to talk a little bit about carbon. So carbon, super, super important atom when we talk about the formation of organic molecules, okay? And so it says there, the carbon atom is small, only has 6 electrons, okay? So it has 2 in the first shell and then 4 in the outer shell. And if we remember what we talked about in the last chapter about that outer shell, or that valence shell, right? That's what's going to dictate how the element or the atom, reacts, right? Or how it's going to form bonds. Okay, so carbon only has 4 electrons in its outer shell. So in order to fill that valence shell, it needs another 4. Okay, so it says carbon can form 4 covalent bonds because of that, because it has those 4 sort of missing electrons, if you want to think of it that way. And so it will form bonds with carbon, right? Another carbon, it'll form bonds with nitrogen, hydrogen, oxygen, phosphorus, and sulfur. And that really, the carbon-carbon bond, it says here, is very stable. That's really going to form the backbone of many organic molecules, okay? So you have these long carbon chains that are called hydrocarbons, and then often other atoms will be attached to that carbon chain, okay? Also, that those carbon atoms can form double bonds, triple bonds, not only single bonds, okay? And also ring structures. Okay, so there's lots and lots of versatility sort of around that carbon atom, again, just really dictated by the number of electrons in its valence shell, okay? So super important atom when we talk about the formation of organic molecules, okay? So it says branches at any carbon make carbon chains more complex. Just like I said, you sort of can think of having those carbon skeletons, that carbon chain, and then there's lots of different groups that are attached to that carbon chain, okay? All right, so I mentioned this, I used this term already, carbon skeleton or carbon backbone, all right? And then I said there's other things that can be attached to that carbon skeleton or carbon backbone. Those other things we call functional groups, okay? So functional groups are clusters of specific atoms that are attached or bonded to the carbon skeleton. And those functional groups have a characteristic structure and a characteristic function, okay? And it's those functional groups that really determine the chemical reactivity of organic molecules and whether they are polar or nonpolar, all right? And so I'll show you a table in a second of some different functional groups. We're not required to memorize these functional groups, but understand the importance of them, and that those functional groups are going to dictate the chemical reactivity of the molecule, okay? So it gives you an example here. It says if you replace a hydrogen by a hydroxide, right, an OH, in a 2-carbon, basically a 2-carbon hydrocarbon ethane. So, right, it's just 2 carbons, some hydrogens. And then instead of one of those hydrogens, you put a hydroxide, an OH on it, it will turn it into ethanol, okay? And it will change that molecule from hydrophobic to hydrophilic. So think for a second what those terms mean. Hydrophobic, right, means that it cannot readily dissolve in water. Hydrophilic can. That's going to dramatically change the chemical reactivity and chemical properties of that molecule. Again, and it's due to this functional group that's added, okay? Or in this case, replacing one of the hydrogens. Okay, so this is showing you here a hydrocarbon, right? Exactly as it says, a hydrocarbon is a molecule that has carbons and hydrogens. So remember, this is the backbone or the skeleton that we're talking about, this sort of carbon chain, and then you have all hydrogens attached, right? Basically to complete carbon's sort of octet, right, in that valence shell, right? Remember, it has 4 electrons, it needs 4 others, and so that's why it can form 4 covalent bonds, right? And so this is just giving you an example of octane. Again, I don't expect you guys to name molecules based on structures or anything like that. That's for chemistry. You'll do that in chemistry. And so I really don't focus on that here. Okay. So like for instance, on an exam, I wouldn't, just show you this structure and say, what is the name of this molecule? I'm not going to do that. Although it is kind of easy. Because you got 1, 2, 3, 4, 5, 6, 7, 8 carbons, hence the name octane, but we're not going to do that, okay? All right, so same thing again. This is also a hydrocarbon. What's different? It's not a straight line, right? Now this is a ring structure, okay? This is cyclohexane. Again, it's only composed of carbons and hydrogens, okay? Hence why it's called a hydrocarbon, but you can see that the structure is much different, okay? Again, why is it called hexane? It has it has 6 carbons, okay? Cyclohexane. Again, we're not going to be responsible for identifying or drawing molecules. That is what you have. That's what you will focus on in chemistry. Okay, so I told you about these functional groups, right? And that the presence of these functional groups is really what's going to dictate the chemical reactivity or chemical properties of a molecule, okay? And so we gave the example of, a hydroxide, okay? And so this is showing you here this hydroxyl group, right? If we call it hydroxide, it's an OH negative, but so anyway, hydroxyl, right? OH. And so basically this is just showing you the R is just representing, a carbon chain or what have you. And then you're seeing these functional groups, okay? Again, I'm not going to say, I'm not even going to say or ask you on the exam, OH, is that hydroxyl, carbonyl, or carboxyl? I'm not going to ask you the name of the functional group or anything like that. Again, that's something that is sort of beyond the scope of this class, and really you focus on it in chemistry. For now, I want you guys to understand that these all exist, right? And that it's the functional group that's going to really change the chemical reactivity of that molecule. And you can see, if you look in this column here that shows you significance, it's going to tell you why, right? So again, that example we gave, if you have if you replace a hydrogen with a hydroxyl group, it makes that molecule an alcohol, okay? And so alcohols are polar, so it would change, our example, we changed a molecule from being non-polar to polar, okay? And so again, you can see, you can kind of take a look at all these different functional groups to see kind of where you find them, right? And then what the significance is. All right. Again, we're not memorizing this, okay? We're understanding that the functional group is going to dictate the chemical reactivity of the molecule, right? And understanding that there are different types of functional groups, all which lend specific types of properties to that molecule, all right? And you'll see some of these will come up as you move on. I mean, as you move on in more of your biology courses, like say in cell bio, we talk about phosphate groups a whole lot, right? And so kind of amino groups, you'll see we'll talk about those again when we talk about proteins later in this chapter. So these sort of come up along the way in this course, but also in subsequent courses as well, all right? Okay, another thing to mention is something called an isomer. And so here, what you see is, again, the sort of the formula, the chemical formula here is the same, right? You have 3 carbons. If you count up the number of oxygens, you have 3 oxygens, right? And if we look at the number of hydrogens, 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. You have the same number of hydrogens, right? So the actual chemical formula is the same, but the structure is different. And particularly what's different is basically where that double bond is in the molecule, okay? And so these are two different molecules, okay? Glyceraldehyde and dihydroxyacetone. Again, not going to ask you to name these structures, not going to ask you to draw any of these structures. These are just meant to be illustrative. So for our purposes now, we want to understand what is an isomer, right? So if I showed you this and asked you about these, we should be able to identify that these are isomers of each other. And again, it has to do with where that double bond, the placement of that double bond within that molecule, okay? It's in a different place. All right. Isomers, if we want to define it, organic molecules that have identical molecular formulas, like I said, right? Identical chemical formula, but a different arrangement of the atoms. Again, if we're looking here, the placement of that double bond is different, okay? All right, so now we will focus on these biomolecules, right? These 4 classes of biomolecules. We're going to talk about each one of them separately. First, though, we're going to talk about biomolecules in general. We're going to talk about kind of some terms that are going to generally apply to all of these different specific groups. We're going to also talk about some reactions that will apply to all 4 classes of these biomolecules, okay? So if kind of what I tell you first right now maybe doesn't sink in or doesn't make as much sense, maybe as it should, it probably will as we get to the end of the chapter, because we will repeat these concepts over and over again specific to each class of biomolecules, okay? All right, so again, reminder, carbohydrates, lipids, proteins, nucleic acids, these are all biomolecules. Those are the 4 main classes of biomolecules, okay? Usually they consist of repeating units, and we call the repeating units monomers. Again, this is general terminology, okay? When we talk about the specific classes, we'll maybe add in some more specific sort of names for these monomers, okay? So general monomer. A molecule is composed of that's composed of these monomers, like many monomers, is called a polymer. Okay, so mono, one, poly, many. Okay, so example, right? Amino acids are the monomers that form proteins. Protein is the polymer. So, like I said, as we move through and talk about these specific groups, we'll add in specific names. So here's the example. Here's one example right here. Again, what is the monomer of a protein? It's amino acids. Amino acids, many amino acids join together to form polypeptides or to ultimately form proteins, okay? The one sort of, I guess, kind of exception here, it says lipids are not polymers because they contain 2 different types of subunits. So technically, you're not going to call a lipid a polymer because it doesn't have the same type of subunits sort of joined together. They're different. But to be honest, the sort of general sort of concept is the same, okay? All right, so that's what this is showing you here. If we look at this little, I mean, obviously showing you a little dinner table there, you know, I guess it's kind of easiest to kind of think about these different types of biomolecules in relationship to food, maybe some of them, right? So if you talk about the 4 categories here, carbohydrates, right? The subunits or the monomer, the reason why we're calling it a subunit, not really a monomer, it's the same difference, right? So many monosaccharides will join together to form polysaccharides, the polymer. And this is just showing you here that these, right, with the stars are technically polymers, but lipids aren't. Remember, I just mentioned that on the last slide. So what are the subunits of, give me an example of subunits of fats, right? So glycerol and fatty acids join together to form fats. Fats are a type of lipid. There are other types of lipids that we will talk about as well. For proteins, we already gave this example. Amino acids are the subunits. They join together to form polypeptides, and then ultimately they'll go through some other folding to become functional proteins. Nucleic acids, okay? Here, nucleotides are the subunits. Nucleotides join together to form DNA and RNA, genetic material, okay? All right. Now, like I said, I'm going to talk about general reactions here that then will apply to all of these different biomolecules. So first one, dehydration reaction. So just as it's named, dehydration, right, means removal of water. Okay, so a dehydration reaction will join subunits together, okay, by the formation of covalent bond. And like I said, water is removed during the reaction. So we said with our example, of amino acids and proteins, amino acids, when they join together, it is a dehydration reaction. Here's another example, right? When glucose subunits, which are monomers, join together to form, say, starch or some other polymer, when they join together, it is a dehydration reaction, okay? So dehydration reactions will join monomers together, okay? And it's called a dehydration reaction because water is removed. Hydrolysis, on the other hand, is the opposite. A hydrolysis reaction occurs when you break a covalent bond. Okay, you have a polymer and you're breaking one of the bonds between the monomers to basically release them and separate the monomers. Okay, so that's what it says here, used to break down polymers into monomers. And hydrolysis, it's called hydrolysis because a water molecule is added when that bond is broken. Okay, so it says here, now we're going opposite here, digestion of starch. So you have starch, which is a complex carbohydrate, which is made up of a whole bunch of glucoses. Okay, when we say digestion, we mean breaking it down, right? So you're breaking it down each time you break the bonds between those glucose monomers or those glucose subunits. It's a hydrolysis reaction. Okay, I will show you a picture right now. Now, we should be familiar with this. We should be able to identify, so this says hydration or hydrolysis is what we said, same difference, right? We should be able to identify, if I show you this picture, which one is dehydration and which one is hydrolysis, right? Or hydration. And which one occurs when you're synthesizing a biomolecule, right? Joining monomers together. And which one is happening when you're degrading or breaking down a biomolecule here, okay? So again, you have two monomers. So if we go back to our example on the last slide, which was glucose, right? So here's a glucose, here's another glucose. Dehydration reaction will join those two glucoses together, right? You see that bond forms there. In the process, water is lost, okay? And that's why it's called dehydration. If we talk about breakdown, now we have our starch molecule, right? And we're breaking it down. So we're going to cleave this bond here. And by the way, this is happening every single place there's a bond, right? We're super simplifying this and just showing you what's happening with one bond, right? So here, hydrolysis or hydration, basically you're breaking this bond, right? And now you have those two separate monomers again. Those are those two glucose monomers. Water is put in, all right? So we should know this. We should understand this. Okay, in terms of, and this is something, we'll talk about enzymes again, much more later on in a couple chapters, but just to mention this now, whenever we're talking about reactions in the cell, they're going to require enzymes, okay? And so these dehydration and hydrolysis reactions that I just outlined do require enzymes, okay? And again, but we're not going to focus on enzymes right now, but we will talk about them later on, I believe in chapter 6. So what's an enzyme? Just so that we know, enzyme is a molecule that will speed up a reaction, right? So an enzyme is required to drive reactions in the cell. Those enzymes are not consumed in the reaction and they're not changed by the reaction, but they are required to speed up that reaction and basically to speed it up to make it biologically possible or relevant, right? Without enzymes, though these reactions may occur at some point, they would never occur fast enough to basically be physiologically relevant, okay? All right, so I actually am gonna stop there. And so for part 1 was sort of the introduction to all the general, reactions and talking about the biomolecule, the classes of biomolecules. Now we will go through each specific class of biomolecules, right? And talk about the specific monomers and polymers that are formed. All right, so part 2 will start here. Thanks.